third week of development, trilaminar germ disc. Gastrulation, formation of embryonic mesoderm and endoderm. The most characteristic event occurring during the third week of gestation is gastrulation, the process that estabhshes all three germ layers, ectoderm, mesoderm, and endoderm, in the embryo. Gastrulation begins with formation of the primitive streak on the surface of the epiblast, Figs 1 and 2a. Initially, the streak is vaguely defined, Fig 1, but in a 15 to 16 day embryo, it is clearly visible as a narrow groove with slightly bulging regions on either side. The cephalic end of the streak, the primitive node, consists of a slightly elevated area surrounding the small primitive pit, Fig. 2. Cells of the epiblast migrate toward the primitive streak. Upon arrival in the region of the streak, they become flask-shaped, detach from the epiblast, and slip beneath it, Fig. 2 BC. This inward movement is known as invagination. Cell migration and specification are controlled by fibroblast growth factor 8, FGF8, which is synthesized by streak cells themselves. This growth factor controls cell movement by downregulating ecadherin, a protein that normally binds epiblast cells together. FGF8 then controls cell specification into the mesoderm by regulating brachyury, T, expression. Once the cells have invaginated, some displace the hypoblast, creating the embryonic endoderm, and others come to lie between the epiblast and newly created endoderm to form mesoderm. Cells remaining in the epiblast then form ectoderm. Thus, the epiblast, through the process of gastrulation, is the source of all of the germ layers, and cells in these layers will give rise to all of the tissues and organs in the embryo. As more and more cells move between the epiblast and hypoblast layers, they begin to spread laterally and cranially. Gradually, they migrate beyond the margin of the disc and establish contact with the extraembryonic mesoderm covering the yolk sac and amnion. In the cephalic direction, they pass on each side of the precordial plate. The precordial plate itself forms between the tip of the notochord and the oropharyngeal membrane and is derived from some of the first cells that migrate through the node in the midline and move in a cephalic direction. Later, the precordial plate will be important for induction of the forebrain. The oropharyngeal membrane at the cranial end of the disc consists of a small region of tightly adherent ectoderm and endoderm cells that represents the future opening of the oral cavity. Formation of the notochord. Prenotochordal cells invaginating in the primitive node move forward cranially in the midline until they reach the precordial plate, Fig. 3. These prenotochordal cells become intercalated in the hypoblast so that for a short time, the midline of the embryo consists of two cell layers that form the notochordal plate. As the hypoblast is replaced by endoderm cells moving in at the streak, cells of the notochordal plate proliferate and detach from the endoderm. They then form a solid cord of cells, the definitive notochord, Fig. 3c, which underlies the neural tube and is a signaling center for inducing the axial skeleton. Because elongation of the notochord is a dynamic process, the cranial end forms first, and caudal regions are added as the primitive streak assumes a more caudal position. The notochord and prenotochordal cells extend cranially to the precordial plate, an area just caudal to the oropharyngeal membrane, and caudally to the primitive pit. At the point where the pit forms an indentation in the epiblast, the neuroenteric canal temporarily connects the amniotic and yolk sac cavities, Fig. 3a. The cloacal membrane is formed at the caudal end of the embryonic disc, Fig. 2a. This membrane, which is similar in structure to the oropharyngeal membrane, consists of tightly adherent ectoderm and endoderm cells with no intervening mesoderm. When the cloacal membrane appears, the posterior wall of the yolk sac forms a small diverticulum that extends into the connecting stalk. This diverticulum, the allantoenteric diverticulum, or allantois, appears around the 16th day of development, Fig. 3a. Although in some lower vertebrates the allantois serves as a reservoir for excretion products of the renal system, in humans, it remains rudimentary but may be involved in abnormalities of bladder development. Establishment of the body axis, anterior-posterior, AP, craniocaudal, dorsoventral, DV, and left-right, LR, occurs early in embryogenesis and is probably initiated during the morula stage of development with the AP and DV axes specified prior to the LR. By the blastocyst stage, the AP axis is determined and cells that are destined to form the anterior visceral endoderm, AVE, at the cranial end OF the endoderm layer OF the bilaminar disc, Fig.4, migrate toward what will become the head region. At this bilaminar disc stage, cells in the AVE express genes essential for head formation, including the transcription factors 0TX2, LIML, and HESXL, and the secreted factors Cerberus and leptal, members of the TGF-3 family, which inhibit nodal, a member of the TGF-3 family, activity, thereby establishing the cranial end of the embryo. The absence of Cerberus and leptal at the caudal end of the embryo allows nodal expression to continue, and this signal establishes and maintains the primitive streak, Fig. 4. Once the streak is formed, nodal upregulates a number of genes responsible for formation of dorsal and ventral mesoderm and head and tail structures. Another member of the TGF-3 family, bone morphogenetic protein 4, BMP4, is secreted throughout the embryonic disc, Fig.4. In the presence of this protein and FGF, mesoderm will be ventralized to contribute to kidneys, intermediate mesoderm, blood and body wall mesoderm, lateral plate mesoderm. In fact, all mesoderm would be ventralized if the activity of BMP4 were not blocked by other genes expressed in the node. For this reason, the node is the organizer. It was given that designation by Hans Spemann, who first described this activity in the dorsal lip of the blastopore, a structure analogous to the node, in ex Oedipus embryos. Thus, CHORD-IN, activated by the transcription factor GUSCO-ID, noggin, and follistatin antagonize the activity of BMP4. As a result, cranial mesoderm is dorsized into notochord, somates, and somatomers, Fig.4. Later, these three genes are expressed in the notochord and are important in neural induction in the cranial region. As mentioned, nodal is involved in initiating and maintaining the primitive streak. Similarly, HNF3/3 maintains the node and later induces regional specificity in the forebrain and midbrain areas. Without HNP3/3, embryos fail to gastrulate properly and lack forebrain and midbrain structures. As mentioned previously, guscoid activates inhibitors of BMP4 and contributes to regulation of head development. Over or underexpression of this gene in laboratory animals results in severe malformations of the head region, including duplications, similar to some types of conjoined twins, Fig.5. Regulation of dorsal mesoderm formation in middle and caudal regions of the embryo is controlled by the brachyury T gene expressed in the node, notochord precursor cells, and notochord. This gene is essential for cell migration through the primitive streak. Brachyury encodes a sequence-specific DNA binding protein that functions as a transcription factor. The DNA binding domain is called the T-box, and there are more than 20 genes in the T-box family. Thus, mesoderm formation in these regions depends on this gene product, and its absence results in shortening of the embryonic axis, caudal dysgenesis. The degree of shortening depends on the time at which the protein becomes deficient. Laterality, LR-sidedness, also is established early in development. Normally, many organs exhibit asymmetries, including the heart, lungs, gut, spleen, stomach, liver, and others. Positioning these organs and establishing their asymmetries is orchestrated by a cascade of signal molecules and genes. When the primitive streak appears, FGF8 is secreted by cells in the node and primitive streak, and this growth factor induces expression of nodal, Fig 5.6a. Nodal expression is then restricted to the left side of the embryo by the accumulation of serotonin, 5-HT, on the left side. These high concentrations of 5-HT on the left activate expression of the transcription factor MAD3 that restricts nodal expression to the left side of the primitive node, Fig 5.6b. Midline genes like sonic hedgehog SHH, LEFTYL, and ZIC3, a gene on the X chromosome that codes for a zinc finger transcription factor, are involved in establishing the midline but also prevent nodal expression from crossing over to the right side. Ultimately, nodal protein in the left lateral P8 mesoderm initiates a signaling cascade that includes lefty2 to upregulate PITX2, Fig 5.6b. PITX2 is a homeobox-containing transcription factor that is a master gene responsible for establishing left-sidedness, and its expression is repeated on the left side of the heart, stomach, and gut primordia as these organs are assuming their normally symmetrical body positions. If the gene is expressed ectopically, example on the right side, this abnormal expression results in laterality defects, including situs inversus and dextrocardia, placement of the heart to the right side, P65. Note that the neurotransmitter 5-HT also plays a critical role in this signaling cascade that establishes laterality. 5-HT is concentrated on the left side, which activates MAD3 and restricts nodal signaling to the left side, Fig.6b. Animal studies show that altering 5-HT signaling can result in situs inversus, dextrocardia, heart malformations, and a variety of laterality-related birth defects called heterotaxy, whereas epidemiology studies indicate that similar malformations occur in humans when 5-HT signaling is disrupted by pharmaceutical agents. Genes regulating right-sided development are not as well defined, although expression of the transcription factor SNAIL is restricted to the right lateral plate mesoderm and probably regulates effector genes responsible for establishing the right side. Why the cascade is initiated on the left remains a mystery, but the mechanism may involve cilia on cells in the node that beat to create a gradient of nodal toward the left or by a signaling gradient established by gap junctions and small ion transport. Fate map established during gastrulation. Regions of the epiblast that migrate and ingress through the primitive streak have been mapped, and their ultimate fates have been determined, Fig.7. For example, cells that ingress through the cranial region of the node become precordial plate and notochord, those migrating at the lateral edges of the node and from the cranial end of the streak become paraxial mesoderm, cells migrating through the midstreak region become intermediate mesoderm, those migrating through the more caudal part of the streak form lateral plate mesoderm, and cells migrating through the caudalmost part of the streak contribute to extraembryonic mesoderm, the other source of this tissue is the primitive yolk sac hypoblast. Growth of the embryonic disc. The embryonic disc, initially flat and almost round, gradually becomes elongated, with a broad cephalic and a narrow caudal end, Fig 2a. Expansion of the embryonic disc occurs mainly in the cephalic region, the region of the primitive streak remains more or less the same size. Growth and elongation of the cephalic part of the disc are caused by a continuous migration of cells from the primitive streak region in a cephalic direction. Invagination of surface cells in the primitive streak and their subsequent migration forward and laterally continues until the end of the fourth week. At that stage, the primitive streak shows regressive changes, rapidly shrinks, and soon disappears. That the primitive streak at the caudal end of the disc continues to supply new cells until the end of the fourth week has an important bearing on development of the embryo. In the cephalic part, germ layers begin their specific differentiation by the middle of the third week, whereas in the caudal part, differentiation begins by the end of the fourth week. Thus, gastrulation, or formation of the germ layers, can't use in caudal segments while cranial structures are differentiating, causing the embryo to develop cephalocaudally. Clinical correlates, teratogenesis associated with gastrulation. The beginning of the third week of development, when gastrulation is initiated, is a high eye-sensitive stage for teratogenic insult. At this time, fate maps can be made for various organ systems, such as the eyes and brain any age, and these cell populations may be damaged by teratogens. For example, animal studies indicate that high doses of alcohol at this stage kill cells in the anterior midline of the germ disc, producing a deficiency of the midline in craniofacial structures and resulting in hoyoprosencephaly. In such a child, the forebrain is small, the two lateral ventricles often merge into a single ventricle, and the eyes are close together, hypotelorism. Because this stage is reached 2 weeks after fertilization, it is approximately 4 weeks from the last menses. Therefore, the woman may not recognize that she is pregnant, having assumed that menstruation is late and will begin shortly. Consequently, she may not take precautions she would normally consider if she knew she was pregnant. Gastrulation itself may be disrupted by genetic abnormalities and toxic insults. In caudal dysgenesis sirenomelia, insufficient mesoderm is formed in the caudalmost region of the embryo. Because this mesoderm contributes to formation of the lower limbs, urogenital system, intermediate mesoderm, and lumbosacral vertebrae, abnormalities in these structures ensue. Affected individuals exhibit a variable range of defects, including hypoplasia and fusion of the lower limbs, vertebral abnormalities, renal agenesis, imperforate anus, and anomalies of the genital organs, Fig 8a6. In humans, the condition is associated with maternal diabetes and other causes. In mice, abnormalities of brachyury WNT and engrail genes produce a similar phenotype. Tumors associated with gastrulation sometimes, remnants of the primitive streak persist in the sacrococcygeal region. These clusters of pluripotent cells proliferate and form tumors, known as sacrococcygeal teratomash, which only contain tissues derived from all 3 germ layers, Fig 5.9. This is the most common tumor in newborns, occurring with a frequency of 1 in 37,000. Teratomash may also arise from primordial germ cells that fail to migrate to the gonadal ridge. Birth defects associated with laterality situs solidus refers to the normal positioning of the internal organs. Situs inversus refers to a condition where the positioning of all organs is reversed in a mirror image arrangement. Discordant organ positioning with respect to symmetry, where one or more organs are abnormally reversed in position, or if isomerisms, i.e., both atria in the heart look the same, or inversions, i.e., the TWO ventricles in the heart are reversed, are present, is called situs ambiguus or heterotaxy. These individuals are considered to have laterality defects that have occurred because of a failure to properly establish the LR axis. Patients with these conditions do not have complete situs inversus but appear to be predominantly bilaterally left-sided or right-sided. The spine reflects the differences. Those with left-sided bilaterality have polysplenia, and those with right-sided bilaterality have asplenia or hypoplastic spine. Individuals with situs inversus do not have a high risk for having other congenital abnormalities, although they have a slightly greater risk of having a heart defect, but their progeny have an increased risk for having laterality defects and an even higher risk for having a severe cardiac malformation. AISO, approximately 20% of patients with complete situs inversus have bronchiectasis and chronic sinusitis because of abnormal cilia, Cartaginer syndrome. Interestingly, cilia are normally present on the ventral surface of the primitive node and may be involved in LR patterning. In contrast, patients with situs ambiguus, heterotaxy, have a high risk of having other birth defects, including a variety of midline malformations, such as neural tube defects, cleft palate, anal atresia, etc. Furthermore, 90% of these individuals will have complex congenital heart defects. The heart exhibits more laterality than most organs, and perhaps that explains its increased susceptibility when the LR signaling pathway is disrupted. Mutations in the zinc finger transcription factor RZ/C3, a gene located on the X chromosome, cause X-linked heterotaxy. These individuals have a variety of birth defects, including neural tube defects, limb abnormalities, and omphaloceal, and most also have severe heart malformations. The association between laterality and midline defects, neural tube defects, cleft palate, etc., suggests that signaling pathways establishing the AP and LR axes must interact to specify the correct positioning of the body's organs and other structures. The neurotransmitter serotonin, 5-HT, is an important signal molecule for establishing laterality, and animal studies show that disrupting 5-HT signaling results in cases of situs inversus, heterotaxy, dextrocardia, and a wide variety of heart defects. Epidemiology studies show that children born to mothers who take antidepressants from the class of drugs called selective serotonin reuptake inhibitors, SSRIs, Prozac, Paxil, Zoloft, Lexapro, Celexa, etc., have an increased risk of having many different types of heart malformations and multiple other birth defects, thus providing additional evidence for 5-HT's importance in establishing laterally further development of the trophoblast. By the beginning of the third week, the trophoblast is characterized by primary villi that consist of a cytotrophoblastic core covered by a syncytial layer. During further development, mesodermal cells penetrate the core of primary villi and grow toward the decidua. The newly formed structure is known as a secondary villus. By the end of the third week, mesodermal cells in the core of the villus begin to differentiate into blood cells and small blood vessels, forming the villus capillary system. The villus is now known as a tertiary villus or a definitive placental villus. Capillaries in tertiary villi make contact with capillaries developing in the mesoderm of the chorionic plate and in the connecting stalk. These vessels, in turn, establish contact with the intraembryonic circulatory system, connecting the placenta and the embryo. Hence, when the heart begins to beat in the fourth week of development, the villus system is ready to supply the embryo proper with essential nutrients and oxygen. Meanwhile, cytotrophoblastic cells in the villi penetrate progressively into the overlying syncytium until they reach the maternal endometrium. Here they establish contact with similar extensions of neighboring villus stems, forming a thin outer cytotrophoblast shell, Figs 12.13. This shell gradually surrounds the trophoblast entirely and attaches the chorionic sac firmly to the maternal endometrial tissue, Fig. 12. Villi that extend from the chorionic plate to the decidua basalis, decidual plate, the part of the endometrium where the placenta will form, are called stem or anchoring villi. Those that branch from the sides of stem villi are free, terminal, villi, through which exchange of nutrients and other factors will occur. The chorionic cavity, meanwhile, becomes larger, and by the 19th or the 20th day, the embryo is attached to its trophoblastic shell by a narrow connecting stalk, Fig. 12. The connecting stalk later develops into the umbilical cord, which forms the connection between the placenta and embryo. Summary: the most characteristic event occurring during the third week is gastrulation, which begins with the appearance of the primitive streak, which has at its cephalic end the primitive node. In the region of the node and streak, epiblast cells move inward, invaginate, to form new cell layers, endoderm and mesoderm. Cells that do not migrate through the streak but remain in the epiblast form ectoderm. Hence, epiblast gives rise to all three germ layers in the embryo, ectoderm, mesoderm, and endoderm, and these layers form all of the tissues and organs, Figs 2 and 3. Prenotochordal cells invaginating in the primitive pit move forward until they reach the prechordal plate. They intercalate in the endoderm as the notochordal plate, Fig 3. With further development, the plate detaches from the endoderm, and a solid cord, the notochord, is formed. It forms a midline axis, which will serve as the basis of the axial skeleton, Fig 3. Cephalic and caudal ends of the embryo are established before the primitive streak is formed. Thus, cells in the hypoblast, endoderm, at the cephalic margin of the disc form the ave, which expresses head-forming genes, including OTX2, LIML, and HESXL and the secreted factor Cerberus. Nodal, a member of the TGFF3 family of genes, is then activated and initiates and maintains the integrity of the node and streak. In the presence of FGF, BMP4 ventralizes mesoderm during gastrulation so that it forms intermediate and lateral P8 mesoderm. Cordon, noggin, and follistatin antagonize BMP4 activity and dorsalize mesoderm to form the notochord and somatomers in the head region. Formation of these structures in more caudal regions is regulated by the BRA-CHU-RE, T, gene, Fig 4a. Laterality, left-right asymmetry, is regulated by a cascade of signaling molecules and genes. FGF8, secreted by cells in the node and streak, induces nodal and left E2 expression on the left side, and these genes upregulate PITX2, a transcription factor and master gene for left-sidedness, Fig.6. The neurotransmitter serotonin, 5-HT, also plays a role in establishing laterality by restricting nodal expression to the left side. Normal LR positioning of the organs is called situs solidus, whereas their complete reversal is called situs inversus. When one or more organs are abnormally positioned, the condition is called situs ambiguus or heterotaxy. Individuals with situs inversus have a low risk of having other birth defects, but their children have a higher risk, especially for heart defects. In contrast, patients with heterotaxy are at a high risk of having many types of congenital malformations, and almost all will have some type of cardiac abnormality. Disruption of 5-HT levels, or misexpression of genes in the laterality signaling pathway, such as PITX2, results in laterality defects, such as dextrocardia, situs inversus, and cardiac abnormalities. Mutations in the zinc finger transcription factor ZIC3 cause X-linked heterotaxy, with severe cardiac malformations and other defects. Epiblast cells moving through the node and streak are predetermined by their position to become specific types of mesoderm and endoderm. Thus, it is possible to construct a fate map of the epiblast showing this pattern, Fig. 7. By the end of the third week, 3 basic germ layers, consisting of ectoderm, mesoderm, and endoderm, are estabshed in the head region, and the process continues to produce these germ layers for more caudal areas of the embryo until the end of the 4th week. Tissue and organ differentiation has begun, and it occurs in a cephalocaudal direction as gastrulation continues. In the meantime, the trophoblast progresses rapidly. The villi obtain a mesenchymal core in which small capillaries arise, Fig. 12. When these villous capillaries make contact with capillaries in the chorionic plate and connecting stalk, the villus system is ready to supply the embryo with its nutrients and oxygen, Fig. 13.